if aging is truly caused by somatic mutations, then that's it. We can never fix that. It's not possible. And we will basically always remain stuck at our maximum lifespan. I mean, I don't know, you may be aware of the fact that we published a paper in Nature in 2016, I believe. Yeah, 2016, where we basically argued that human lifespan has a natural limit of 115 years. And we basically derived that from demographic studies on existing data sets. There was an enormous conversation about that. Because a lot of people were very disappointed. They couldn't believe it because they were so optimistic that we could begin to increase human lifespan because there's so much talk about neuroscience and everything. And now we sort of threw a bucket of cold water over that by saying, well, forget it, because this is it. I mean, there's absolutely zero evidence that we have improved human maximum lifespan. Yes, we improved mean lifespan. Obviously, we did. People have much more chance now to grow old and stay healthy than in the past. And maybe we can still improve on that, although even I have my doubts about that too. But definitely we cannot break through the limit.
Hello, Dr. Vijg. You are a professor and the chair of the Department of Genetics at the Albert Einstein College of Medicine. So welcome to Modern Healthspan, and thank you so much for joining us today.
Sure, you're welcome. Happy to be here.
Thank you. So, Dr. Vijg, so in your work, you look at genome instability and its impact on aging. And so to start with, could you define what is genome instability?
Right. Well, as nowadays most people probably know, your genes, that's part of your genome. The genome is very large, of course. It's 3 billion building blocks, actually 6 billion if it's a diploid genome. And it contains genes and lots of other codes that sort of influence those genes. How active are these genes, or are they active at all? Now, genome instability is when there are changes in that code, in that sequence code, as we call it. And those changes occur. That's a natural thing. I mean, it's very difficult to replicate such a genome from cell to cell. When cells divide, they have to replicate the whole genome. And you can already imagine in germ cells that have to give rise to the new generation, that maybe that's even more complicated. So there are definitely errors there. But also there's a lot of damage to DNA, physical and chemical damage. There are breaks continuously happening because there are so many agents that you're exposed to through food, through the air, through everything. There's radiation, for example. But even if you're not exposed to radiation, there's always a little bit of background radiation. You cannot avoid it. And think when you're going to the dentist and you get an X-ray. It's very little, of course, but altogether it does add up. And then there are the very natural agents in your body. We call it free radicals that are a normal byproduct of living, of breathing and using energy. And those, what we call free radicals, there are very low concentrations, of course, but still they can cause breaks in DNA and other damage to the bases, and they need to be repaired. Now, in fact, there's a lot of damage. We now know that there may be 100,000 lesions, as we call it, so a break or some sort of adduct that damage the base per cell per day. That's enormous. So you would really drop that very quickly were it not for the fact that we have fantastic DNA repair systems. They very quickly cut the damaged part out of the genome and put a new one in. But you can imagine that also errors happen there. So all these errors during the replication of your genome, but also errors during the repair of it, they make changes in the code, random changes in the code. That's what mutations are. And that is the process that we call genome instability. And some of these mutations are very small. There's one base pair, one wrong base pair for a correct one. But there can also be very large ones. For example, when there are double-strand breaks in the DNA, so both strands of a DNA molecule are broken at the same point, or more or less the same point, and there are multiple of those double-strand breaks in a cell, then the wrong ends can ligate to each other. And now you got sometimes a very big change in your DNA, and that has impact. Now, we know that has impact because very long time studies now have definitely proven that cancer is a disease caused by mutations. There's an accidental mutation in a gene that is involved in the growth of a cell, and suddenly that cell begins to grow while it's not supposed to do that. It could be a cell in your lungs or so that is supposed to give function to your lungs and sit there, but now it gets this mutation in a gene. It changes the characteristics, and there you are. You get the beginning of a cancer, and that's how cancers arise. Now, there are many other age-related diseases, of course, and we do not really know if they are also caused by mutations, but there is now more and more evidence that that could actually be the case. And there's also some evidence that the normal aging process, so the slow degeneration of function, slow decline of function and degeneration of all sorts of processes, that that could also be caused by DNA mutations. Now, that's not a new hypothesis at all. In the 1950s, that was proposed. So only a couple of years after the DNA molecule was discovered by Watson and Crick, then it was already proposed, oh, so this is what mutations are, because now we know how genes look like. We never knew that. People guessed it could be proteins, it could be anything, but then they knew how it was organized. This is DNA, it's in the nucleus, in each cell, and also in germ cells. So that is how your characteristics, your personal characteristics are being transferred from parents to children, and also how cells give function. So then smart scientists at the time realized, hey, but we know now that mutations occur. We also knew that already earlier, actually, when we didn't even know what DNA was. We did know that there were sort of heritable changes occurring randomly. For example, in animals exposed to radiation. We didn't know it was DNA, or I say we, but I was of course not present there, but it was hypothesized that this was well, a genetic change that could occur. And then when Watson and Crick published the DNA molecule, they thought, oh, but that is what it is. There's just a mutation, a change in the DNA. And these people thought, but that can happen in all your somatic cells. And that probably accumulates during the aging process. So that must cause the aging process. So this is basically the first time that people began to hypothesize that aging itself was caused by mutations. And then later, people found out that cancer was indeed caused by mutations. Cancer is an age-related disease. Although young children, of course, as you know, as most people know, can get cancer. It's a very sad story, but it happens. It's actually very rare. Cancer is a disease of the old. So it's not difficult to then hypothesize, look, there's this continuous accumulation of mutations in your somatic cells. You cannot avoid it. Errors are errors. You cannot do anything about it. Well, maybe we will come back to this later in this discussion, maybe, but at the time it was not considered possible. So now you have this inevitable process of mutation accumulation, so ongoing genome instability, and that now gives rise maybe not only to cancers, but also to the loss of function and all sorts of other age-related diseases like heart disease. So that's essentially the background story of this.
A couple of things to dive into there. So one thing, just to summarize, so DNA damage is not the same as somatic mutations, so mutations
Correct.
in the DNA.
Entirely correct. That is a mistake often made also by scientists, by the way. So it's a very common mistake.
So DNA damage is it's the damage to the genome that happens. And then the mutation is if that damage leads to a change in the base pairs.
That is exactly right. And the mutations are caused by errors made during copying the DNA, during cell division, or during the repair of DNA damage. Exactly.
Okay. So the thing that you look at are the mutational changes or the DNA damage or both?
Well, now already for a very long time, I'm only focused on the mutations. But when I was a student, I did a lot of work on DNA damage as well and the repair processes. But now we are focused entirely on the mutations. And that was when I started this research, which is now a very long time ago in the 1980s and 1990s. then there were no methods to measure mutations. I mean, DNA damage, we thought we could measure it. I say we thought because it turned out to be very difficult too. But mutations, we thought there's no way to detect those because they're very rare. And they occur, of course, they are different from cell to cell. So if you take like a piece of liver or you take a blood sample and you extract DNA from bulk, you will sort of, it dilutes out. You will not see all those mutations, although there can be many mutations. They are all different from cell to cell, so you will not see them. There's no way. Now, we developed the first methods that allowed people to actually did look at that. That was a mouse model that we developed. I'm not going into detail here, it's not so important, but we always wanted to do this directly. So take a human cell sample, blood sample or liver sample, extract DNA from it, and then measure the random mutations that occur during aging. And to do this, we developed single cell methods. So because I just said mutations are rare and they differ from cell to cell. Say one cell can have thousands of mutations, but another cell has thousands other mutations. They're not the same. They can be the same very rarely, but normally never are. You can imagine, I just mentioned the genome is a total of 6 billion bases long. So mutations, there are many opportunities for mutations. So the chance that two different cells get the exact same mutation at the same spot is very, very small. So it tells you how difficult it would be to measure it. But when you take a single cell, you will find all mutations. When you can sequence the genome of that single cell accurately and compare it to the bulk genome, so to say, then you got it. You find a way to do that. Now, that is obviously very, very difficult, and I'm not going to go into detail, but we managed to develop that, and other people then also. And so then we began to sequence multiple cells from human liver, from human lung, from human bone, from blood, from skin cells, and then basically take those samples from young people, mid-age and old. And we could see that in all those cases, we could see that mutations go up. In a, say, 1 day old individual, a newborn, the mutations that we found were a couple of hundred per cell. And I'm talking about small mutations now, like one base pair. But if you looked at, for example, liver cells of a person in the 70s, we could see 8,000 of those mutations per cell. So there's a pretty dramatic increase in the mutations. We, in a way, knew that from our mouse model, because in the mouse we found a very similar thing. But in the mouse, we could never sequence a whole cell. We couldn't do that. It was an indirect system, a selectable marker gene, as we call it. It's not so important here. But now in humans, we could, for the first time, really see that very clearly, that all those mutations accumulate. But now, of course, the next question is it enough? Can that really explain the aging process? That's sort of the big question now.
Yes, and kind of where are you on that? I mean, on showing causality between the increase in mutations and aging.
Right. Well, the evidence thus far is only indirect. The fact that cancer is increasing exponentially with age and mutations also increase is striking, obviously. But again, there's not a proof. So you first need to have like evidence that mutations can really cause age-related functional decline. Now, there are what we call clonally amplified mutations. I should mention that because that's now really the focus of a lot of studies. So when there is an accidental mutation in a cell, but for some reason that mutation gives the cell a growth advantage, a little bit like in a cancer, but not really. It's not a cancer, but the cell can expand. It can also be simply by chance that cell has daughter cells and they stay alive, and then they have daughter cells also. So they all inherit the same mutation. So now you get a situation where in a tissue, maybe 30%, maybe 10%, maybe 20% of all cells contain the same mutation. Now, it depends what that mutation is doing, but that mutation could have some sort of adverse effect. And now it is a significant part of the tissue. So it could mean that that part of the tissue is dysfunctional. And that, of course, could be a cause of aging. And there is some evidence now that has been found in certain diseases of aging, like in certain liver diseases, but we're still at the beginning of this. So that's sort of the first thing. That's what we call the low-hanging fruit. Now, a more general hypothesis, we proposed that a couple of times and found some evidence for that. That is that when you begin to flood the genome with random mutations, then there's no obvious effect of that because the cell is still a cell. It still looks more or less the same. It's doing what it's supposed to do, but it's not doing it that efficiently anymore. Because there are so many mutations, they begin to adversely affect the way genes talk to each other. Every cell has to perform a particular function. A neuron has to help you thinking, of course, not. It has to channel all those signals to all sorts of somatic cells that need to become active then. And you got blood cells that need to help you in fighting infections and so on and so on. So those, in order to carry out such functions, you cannot do with one gene. We have probably like, well, maybe 20,000 active genes. Genes can potentially be active. And in every differentiated cell, maybe, I don't know, 20%, maybe 30% of those genes is working. In each tissue, there are different genes active because, of course, they have their own function. Not now, how do these genes work? Well, they have to talk to each other. They are basically not on an island, and there are many other parts of the genomes that give them signals that help to activate or deactivate them. Probably in every differentiated tissue, in a lung cell, or in a kidney cell, maybe 10% of the whole genome is important. We always say, or some people say, well, most of the genome is useless. Well, that's probably true. But still, as much as, say, only like 1% of all of the entire genome are genes. But maybe 10% are codes, sequences that are important for those genes to give function. Now, you can imagine, therefore, that if like thousands of mutations begin to accumulate, then occasionally they will hit part of that 10%. That's unavoidable. So it means they may influence maybe not the genes themselves so quickly because it's only 1%, but certainly they will have an effect on how these genes are influenced by all these regulators, how the cell therefore will function. It will not totally inactivate a function, so the cell will not immediately die off. It will still be there and it's still functioning, not as good anymore. Now you see how that looks like aging? This is exactly what aging is. Aging is not like people suddenly when they're 52 years old drop dead. No, you see it gradually degenerating. A good example is temperature control. In the heat wave in France a couple of years ago, there was the story of older people. They drive home or take a taxi home. They get out of the car, walk to their front door. Suppose it's a big house. It's a little far, so they have to walk for a couple of minutes. And there's a heat wave. So can they not regulate the temperature? Yes, they can, but much slower than younger people. This is very well known. Suddenly they drop dead because of that. They cannot maintain their temperature, cannot lower it anymore. Now, temperature control is very complex. It's determined by multiple cells, multiple genes, and therefore multiple regulatory interactions. So you can see how even subtle changes there that you say, well, it's not that bad, is it? No, but it can be enough to reduce your function marginally, and now you are much more susceptible. That is aging. You see it in many functions. Think of the immune response. Immune response still works at older people, not as good anymore as in younger people. So this kind of models, as we now put together, can definitely explain aging. But now you will ask me, have you any proof that that's the case? No, we don't. And that should also be sort of a warning signal. Science is often hyped. I can understand why that is, of course, because people want to get more money for the research, but we should not exaggerate the progress that we make. Progress is really slow, and it should be slow. I mean, these are not simple problems that we're dealing with. I mean, come on. I mean, this is, in essence, the question of how do we age? Well, I mean, we are studying that for thousands of years, in a way, not? I mean, we always try to overcome aging. So that's not a simple question. So don't expect us to know the answer next year or in 10 years or in not maybe not even in 100 years. But it's not that we don't make progress, we make a hell of a lot of progress. I mean, look at it, look at the fact that now we can look very accurately at all those mutations in single cells. And even in single molecules now, we have even more advanced methods. So we can do all these things. And now maybe, I don't know if you want to ask me, can we do something about it? Let's assume that mutations are the cause of aging. Is it then possible to do something?
We will definitely get there.
Okay.
So
I'm sorry, I don't want to feed you the questions, but I personally, you can already imagine that's of course a major issue for us, very enormous
yes,
interest.
absolutely. Does the speed, does the rate of mutation change also increase with age? So obviously over time, like the number of mutations increases, but does the rate increase as we get older?
Yeah, that's a very good question, and it's obviously something we'll try to look at immediately. If you look at our papers, and I don't ask you to do that, but you will see that we have all these different individual cells from young people, from old people. Now, if you look at it, you can clearly see that on average, there's this increase of mutations with age. But if you look at it a second time, you see a cloud of determination points. So all these cells are different from each other. What we were looking for is exactly what you say. We were sort of hoping, well, hoping is not a word, I guess, but sort of expecting maybe to see that the increase would be exponential, very similar to what we see in cancer, not? So in other words, it's not at the beginning it's going very slowly, but then you see it quicker, and quicker. And that could be explained by the possibility that you lose the capacity to repair your DNA. So if repair becomes exponentially worse, then you can imagine there are more errors being made and you get a higher mutation rate. So the mutation rate could increase with age, exactly as you asked me. Now, do we have clear evidence for that? Well, the answer is no, because of this enormous variation. But technically speaking, when you publish papers, people would ask you the question, is this a linear process or is it exponential? And the answer in at least one or two of our papers was that it was indeed exponential. So it seemed to be that the mutation rate does increase with age, which would be in keeping with people who started to look at DNA repair processes and the activity of it. I actually did that in the past also, and showed that DNA repair could actually also be less in older people than in younger people. So it would fit that hypothesis. But again, the answer to that question is still not completely clear.
What are the major causes of mutations? Is it just failure of DNA repair, or are there other reasons?
Yeah, well, there are two main processes. One is simply cells have to replicate, they have to divide. So you have to copy the genome in every cell during every cell division, especially early on during development, of course. There's a lot of cell divisions going on. And therefore, there must be also a lot of errors being made. And you must see a lot of mutations occurring early on. Now, there's some evidence that that's the case, but my assumption is that the early embryo must have a lot of safeguards to prevent that. So maybe what it is doing is it allows all those mutations to happen because cell division is important. There must be, all the tissues must be generated. But then maybe it has like processes that tend to kill the cells that have too much DNA damage as sort of a precaution, because let's assume then that initially when multicellular life emerged, that process was not in place, and then mutations were accumulating so fast that basically the animals died. So that's a very quick selection, not because they die. So now there was an accidental mutation in the germ cells then that gave them genes that suddenly kick in that self-killing process when there's too much DNA damage. And suddenly more cells could survive without mutations because the cells potentially with the most mutations that have had all the DNA damage were killed off. So I just give you an example of how that could work. So during development, therefore, mutations definitely occur, but clearly not that many. Otherwise, we would have seen it. I mean, so that's not the case. Now, again, during aging, as I just said, there is evidence that DNA repair may get worse, and therefore there could be more mutations. That's the second cause of mutations. The first is basically natural errors when you have to copy genomes, and the second is errors during the repair of DNA damage, and that could also change with age. So those are the only two causes of mutations. Theoretically, you could argue there is well, not theoretically, actually, practically, there are cases where whole chromosomes are lost. Now, those are not errors during replication, but they are errors during cell division because chromosomes have to be segregated when a cell divides. And when mistakes are being made, then by accident, they're not neatly divided over the two daughter cells, but one gets two and the other gets nothing of a particular chromosome. So you get a process that we call aneuploidy. So you lose a chromosome or you gain one, which happens during somatic cell division, but also during germ cells. Think of Down syndrome, for example, when you have three copies of chromosome 21, there you have such an error in germ cells. So that's also a mutation. That is actually a mutation. Also, another very well-known chromosomal mutation is loss of a Y chromosome. Many males, not all of them, I guess, but many of them, they lose a Y chromosome in their blood. That was shown already a long time ago because we are able to analyze chromosomes already for a long time, since the 1960s. So it was already noticed that in bone marrow cells or in blood cells, you can see that in males. They lose Y chromosomes. And now apparently you would argue, well, a Y chromosome is not that important, is it? So that's why they can do without one. Well, apparently they can, but because it's a very common age-related effect. There is, by the way, evidence that the loss of a Y chromosome in blood, when you measure that in males in blood and you look at them aging and getting diseases, then the more often you lose Y chromosomes, the higher the mortality rate and the higher frequency of certain diseases, including cancer. So there is some sort of importance here in this kind of mutation. It's just a mutation. It's a whole chromosome. I said you can have not only base pairs, but the whole parts of chromosomes or even whole chromosomes. And so there is some sort of correlation between the level of genome instability in somatic cells and the mortality rate. So the more of this chromosomal or genome instability there is, the higher the mortality rate. Now, we are studying it. We don't really know what it means and what the mechanism is behind it. Why is it like that? We can speculate, but
How does the DNA repair system change with age? I,
yeah,
yeah, how does it function? And also, I believe it takes a lot of energy, right? So,
that's
is the
exactly right. Yes, yes, it does. First of all, there are many DNA repair pathways. You're not talking about one simple repair pathway. It's very complex. And there are probably many, many genes that are important to have a good repair system. So life, of course, considers that extremely important. This is also why it's highly conserved. So you see more or less the same, well, not the same, but very recognizable repair processes in bacteria and in humans, and they are really surprisingly similar. So if you go back in evolutionary history, then DNA repair was probably one of the first actual systems, traits that was selected for it because you needed it. Now, to some extent, you can sort of understand that because the very early life forms, mainly protocells, we call them, they essentially had very small genomes. You're talking maybe probably even about an RNA genome initially, but it's just very small. But you cannot pack a lot of information there. So those were very simple cells. But when evolution continued and there was continuous mutation in natural selection, they created, well, it's the wrong word, but by accident it emerged more complex life forms, and they had larger DNA molecules that carried the information. Now you can imagine what's going to happen when there's a lot of DNA damage. Well, you cannot maintain that. So DNA repair kicked in very, very early to make sure that actually could work. So this is why so much energy, as you said, is spent on maintaining your genome. And that in turn is, of course, because, and I said that at the beginning of this discussion, there's so much DNA damage. People always think that DNA must be a very stable molecule, but we now know that that's not the case at all. DNA is not stable. It's very suitable as the carrier of genetic information. Otherwise, it would not have been selected for that purpose. But it is unstable under physiological conditions, breathing, higher temperatures, lots of DNA damage. But we have DNA repair. And as I said, that was there from the very beginning. So now you can fix all that damage very, very quickly in minutes sometimes. When you're talking about human DNA repair, you're talking about many different processes. You have, as I said, different types of damage. Most of the damage is fairly small. It's like a single strand break, or there's a little base damage. And then a process which we call base excision repair, which by itself is already very complex. It's controlled by many, many genes. But it kicks in extremely quickly and more or less in seconds, and certainly in minutes, the repair process is almost completed. Now, there are other processes as well. There is what we call nucleotide excision repair. For example, if you're at the beach and your skin receives all that sunshine, it creates like fusions between two adjacent bases in your DNA. We call that thymine or cytosine dimers. They link together, which is not good. And it gives you like a bulky show. It shows as a bulky adduct in your DNA. So your enzymes can recognize it. You have specific enzymes that monitor the genome continuously for such irregularities. And it picks it up immediately. It cuts the area out and put new one in. We call that nucleotide excision repair. But that can take hours before that's completed. That's the slowest repair process we have, actually. But that's ours. So you can already imagine. Now, that's the second major process. There's another one, double strand break repair, probably very, very important because double strand breaks are very toxic and you can have multiples in a single cell. So here you need a very complex process that uses the other genome. You have two genomes in a diploid cell, it's all in duplicate. It uses the other one as a template to correct it, because here's the important information. As long as you have a diploid cell, you always have the other chromosome as a template. So when there's damage in one, you have the other one that can help you to know what the original code was, so you can fix it. That is why mutations, once they have occurred, are irreversible, because you lose the original template. Once the mutation is created, the error is created by a repair process. It's now continuing to be there. It's copied and copied and copied. You lose the original template, how it originally looked like. Now, even this we do not know for sure. Theoretically, you can argue, well, cells are smart. Maybe they have some sort of template hidden. Now, that's a little bit of a fantasy story, in my opinion. But I just say it because just to underscore the complexities of what we are talking about here. It's not that we know everything. I think we only know a fraction of what there is to be known here. So here we are, and now I only mentioned a few of the DNA repair process, probably the major ones, but there are many others. So now, how they do during aging? Well, you can already imagine when in some way your enzymes, your repair enzymes, age themselves. In other words, undergo changes because of mutations, not when there's an accumulation of mutations, they will also affect the repair processes themselves. Then, of course, that could explain how repair really gets less with age. If you ask me what I think, I would say, well, it seems highly unlikely that repair processes will decline a lot during aging because they're too important. if you have a major problem, you can already see that if one repair process is defective, uh, for example, I just explained the UV repair process, nucleotide excision repair. There are diseases, we call them xeroderma pigmentosum. Those are patients where they were born with a defect in nucleotide excision repair. Now, if that happens, they cannot repair these ultraviolet light-induced lesions, and they get skin cancer so terrible that they die very, very quickly because of skin cancer. Now, of course, we know all that. So they are protected. They never go outside and they are never exposed to the sun. So they can now live quite a long time. And what we now see is that they develop age-related symptoms, including neuronal problems. Now, this is also true for other, because there are many other human diseases that are caused by a repair defect. So this underscores your question. DNA repair is super important. If you really have only one problem, one pathway, you have a terrible disease. If you're lucky, because if you're not lucky, you're already dead. You cannot survive. But they give rise to diseases. There are also cancer processes. People get a lot of cancer. There's a colon cancer type also caused by a defect in a DNA repair pathway. So you get cancer very, very early on as a child and you die. So repair is really terrifically important. And when it is defective and you do survive, you have a disease that mimics the aging process. Now, that is not an argument that aging is caused by mutations, because it could, but it is an argument that aging is, in this case, caused by DNA damage. DNA damage, as we always say, could very well be the underlying main cause, main universal cause of aging. I'm always hesitant to ascribe aging to one cause because it's not very likely to be the case. There are lots of processes that would occur anyway and that may have nothing to do with DNA damage. But if you want to define one factor, one process that is really universal in life and could be a universal cause of aging, that is actually DNA damage, in my opinion. DNA damage can then give rise to mutations, cause cancer, other diseases. It can also maybe affect transcription, so the speed at which the enzymes are reading your genes to create proteins. So you asked that also, is there any evidence that that may decline with age? And yes, there is some evidence that is the case. One of my friends and collaborators, Jan Humakers in the Netherlands, he's actually famous for that. And he noticed that there's an accumulation of DNA damage. I'm not talking about mutations now, but the actual DNA damage. There's a gradual, slow accumulation of DNA damage. And you can see then that the enzymes that transcribe read the code to make first messenger RNA and then protein are actually slowed down a little bit. And that's also not good for you. So you see, this is another example of how complex this whole DNA damage metabolism, as we call it, is.
Do all the different tissues get DNA damage? I will get mutations at the same rate, or is it kind of different?
It's an excellent question. That's true. We don't know. The short answer is we just don't know. But you can imagine, and we sort of conclude that in a sense from the fact that we see very clearly tissue differences in mutation accumulation. We see mutations in humans, I'm not talking about, using our single cell assay. We see mutations accumulate the fastest in human liver. They accumulate less fast, but still relatively fast in human lung. And then they accumulate much faster in the lungs of smokers than in non-smokers. We published that in a paper 2 years ago. So it's very clear. Now, if you look at blood cells, for example, they still clearly accumulate mutations, but much slower than in the other tissues. So there are clearly tissue specificities. What does it mean? Now, different possible explanations. Liver may accumulate mutations to a high rate because it's a detoxifying organ. So when you, for example, when you have, let's say, call it bodily unfriendly molecules, like drugs, for example. Not, drugs you're not supposed to have. So they always are detoxified, as we say it, in the liver. The liver detoxifies anything. Whenever you got something bad, then it's detoxified in the liver. But the liver, therefore, creates also DNA damage quite a lot. So that could explain why we see the most DNA mutations accumulate in the liver, because there's most DNA damage. So there must be a lot of DNA repair, there must be a lot of errors, and there you go, there you have more mutations. Now, but that's also a little strange then, because why is the liver cell then better capable to sustain so many mutations than the blood cell? Well, okay, the liver cells, they tend to become what we call polyploids. So they multiply their whole genome. So rather than only one genome, they have two. They have, of course, they always have two because they're diploid, not? But now they are not diploid, they're tetraploid. So they double it. And sometimes even octoploid. So, so you can hypothesize that is done by evolution, or evolution selected for that, because now the mutations, the effect of the mutations are not so severe. Because now if you have a mutation, then only part of your genomes is affected, not because you still have a lot of clean copies left. So that may be the reason that liver is polyploid. You would say, why are they doing that so complexly? Because they could, in fact, simply select for better repair process. Yes, they could, but maybe it's cheaper, evolutionarily speaking, in terms of energy, to basically create doubling your genome. So maybe that's why the liver did it. So now the blood. In the blood, mutations could be, blood cells could be very important, because what happens in blood is a little bit like what I explained about temperature regulation. Blood cells, lymphocytes, they have to control your immune response. That's a very complex process that requires many, many genes to interact with each other and do signaling. So this is very complex. So when there are too many mutations, and I sort of gave the example earlier on, when you flood your genome with mutations, you may begin to affect that quite severely, all those complicated processes, and you may interfere with that. So maybe evolution simply selected for blood cells to have a very rapid mechanism to kill cells whenever there's too much DNA damage. Because when there's too much DNA damage, there will also be too much mutations, because mutations are caused by errors during repair or replication. So now these blood cells, they protect themselves in that way. And we see, therefore, a slower accumulation of mutations. Is that true? I don't know. We just basically speculate that this is the case. But it is not so unlikely, because we do know that lymphocytes have a very easy trigger for what we call apoptosis, programmed cell death. So indeed, programmed cell death, self-killing of your cells, is more frequent in blood cells than it is in liver cells. Is that a proof of what I just hypothesized? Not really, but we're still getting close. It's always difficult in science, because people always want hard proof of that, and we know exactly how to do it because we have been trained how to do that. But on the other hand, there are very complex biological processes that simply escape relatively easygoing, foolproof ways. Evolution theory is one of them. People always argue, well, there's a lot of evidence against the evolution theory. Actually, that is actually true. You could argue that. However, we are totally convinced that the evolutionary theory is correct. Why is that? Because there's simply too much evidence in favor of it. Such an enormous amount of evidence that there's no way it could be any others. All those arguments against it, you can easily bring those back to particular exceptions. And biology is not physics. Physics is a very hard process where everything is called into iron laws. Biology is extremely flexible. It's almost impossible to catch biology in laws that are always applicable. There will always be exceptions. And this is what you see. There are always exceptions that depend on the environment, it depends on the species, it depends on the conditions. So you need to think about that. So when we build our theories, our first an hypothesis and then slowly our theories of how things work, then that's also a very good way of doing it. So you begin to accumulate more and more and more evidence. And now you can say, well, can we now come up with one simple experiment to prove it? No. Why not? Because the whole story is too complex. There are always exceptions. So you do need to fill the jigsaw puzzle in gradually, and then you, at some point, like we know for the theory of evolution, you know there's no other explanation. This is it. But again, when people ask you questions and they say, yeah, but how about this and this? You say, yeah, that's true. But you say you can do this and this and this. So the discussion begins. But overall, this is a matter of experts. And I'm also not an evolutionary biologist, but I read enough about it to know that that's the story, and we all know it. Aging is like that too. Aging is like a similarly complex process. So also, in my opinion, this is how we will resolve aging, by accumulating so much evidence that at some point we just know what it is, and now we can do something about it.
Is there evidence that improving repair or protecting the genome better in some way extends health span? Like, has there been mouse trials or something?
It's an excellent question. Uh, well, yes, there is evidence. Uh, we actually we just published, it's mostly my colleague Vera Gorbinova. We were the three senior authors on the paper, so I collaborated very closely with her, but she's really the one you need to go to when you want to ask questions about it. But the paper just came out where we provide evidence that the longest lived species we know, well, one of the longest lived species we know, the bowhead whale, is living so long because it has such a good DNA repair system. Now, is that a proof that that DNA repair is the system that controls aging rate? No, but it is very tempting. And again, when you look at, for example, mutation rates, and we've done that in cells, in somatic cells of different species with different lifespan. If you compare, for example, the mouse, which can live in the lab for about 3 years, and we look at the naked mole rat, also a rodent, but it can live like 40 years, 40, then we see that there are much fewer mutations, spontaneous mutations in the naked mole rat than in the mouse. And when you do a treatment, you take naked mole rat cells, you treat them with a mutagen, and then you see how many mutations are induced. There are only few mutations induced. Well, in the mouse, there's a flood of mutations induced. So it tells you that the repair process in the naked mole rat is much better than in the mouse. Others, the group in the United Kingdom, the Sanger Center, Inigo Martin Corina, he's also working on somatic mutations. Good person, by the way, to interview too. He knows quite a lot. He published a very good paper in Nature showing this on a much larger scale. He looked at many different animal species of different lifespan, and he makes it very clear that mutation rate is slower in long-lived species. So that evidence we do have, but it is, as you will say, a correlation, not? So it's not causal. Causal is if we can show that can we upregulate repair and then show that a mouse is now living not 3 years but 6 years. Can we do that? That has never been done.
So is there any interventions that exist now or that people are working on that do help to preserve the genome?
Well, yeah, the obvious ones are, of course, I just gave you the example of smoking. If you smoke, your lungs are exposed to a whole bunch of mutagens in tobacco smoke. And as I just told you, we see a lot more mutations in the lungs of people who smoke. So that tells you when you don't smoke or don't expose yourself to other mutagens and try to use a diet that some people would argue can limit DNA damaging agents like free radicals. I'm very skeptical about that, by the way. But some people would make that argument, then that's sort of low-hanging fruit. But that's not answering your question, and that's also not what's going to do it. Now, people try desperately to begin to overexpress genes involved in DNA repair. But I already mentioned there's so many genes involved in repair processes. So now you begin to overexpress one gene. Suppose the gene that will recognize these bulky forms in your DNA that indicate like a piece of DNA damage, like this UV-induced pyrimidine dimers. So those enzymes are now expressed to a higher level. So they begin to cut very quickly now. Whenever they think, I see something, bang, they cut it. Not good, no? You get more DNA damage, not less. Now, there are many of such examples that when you do that, you begin to overexpress individual repair genes. Effect is actually negative, or the effect is zero. You can understand why that is. Again, I said, there is nothing like one DNA repair gene. There are many of them, and their activity can vary depending on the organ or tissue. It can vary depending on the age level. It can vary lots of things, environment, whatever it is. So that's not going to help you do it. What we need is a master regulator, some sort of magical gene that is going to control all these repair processes. So if you can manipulate that master controller, then DNA repair will become better or worse, of course. Now, very recently, evidence had emerged that that is actually the case. We have something like that. I sort of say that because I just got an email from work where we got some results showing that that actually may be the case. I will come back to that in a second. Now, there's a complex which was discovered a couple of years ago, and it was discovered not because it had anything to do with DNA repair, but it was involved in cell cycle, which is also relevant for DNA repair. So it can control if cells are cycling, so dividing, or when they are not doing that. Now, we call that's a very complex, big complex of proteins. We call it the DREAM complex. It's an abbreviation, DREAM, but we call it obviously DREAM. It's a beautiful name. So now one of my good friends and collaborators in Germany, Björn Schumacher, published a paper in one of the Nature journals a year or two ago, where he showed that that dream complex in worms, worms is his favorite model organism, when he is basically inhibiting that complex, then the repair processes are going up, they are increased. He sees that the expression level of many different DNA repair enzymes is now going up, increasing in tune. They're all just doing the same thing. So it seemed that DREAM is sort of a master regulator here. Now, he also looked at the germline. Now, in germ cells, DREAM is not active. It doesn't work. So that seemed like strange, but it isn't. When DREAM is off, you have very good DNA repair. So germ cells are known to have very good DNA repair. Now, we compared the mutation rate in the germline in comparison to stem cells, in comparison to differentiated cells. The highest mutation rate was found in differentiated cells. In the stem cells, we have not published that yet, well, a little bit in liver, we see lower mutation rate in stem cells. In germ cells, the lowest. There's still mutations, but of course, we know there are mutations in germ cells. That's how evolution works, not? But it's about 10 to 100-fold less than in differentiated cells. Now, you can sort of imagine why that could be, not? We cannot have too many mutations in the germline because otherwise the species will become extinct. So the germ cells must be protected very aggressively, and they are. And DREAM is off. So we can conclude then when DREAM is off, that's good. But we know that in differentiated cells, DREAM is on. I all the time simplify here a little bit, so
Yeah.
but I have to do that. Now, what happens now if you then, as Bjorn did in the nematode cells, when he begins to inhibit DREAM in the differentiated cells? So he makes the differentiated cells a little bit more like germ cells. He sees DNA repairs going up. So the differentiated cells, the normal somatic cells, become more like germ cells. So they have a better repair. And he now knows also, I don't think he has published it, but I'm sure he will not be angry with me when I tell you that he did find that these worms now are indeed longer lived. It's a worm, so it's very difficult to extrapolate to the human, but nevertheless. Now, we work very closely with him. So our reasoning is this. When he now made a mouse model, by the way. In the mouse model, he switched off DREAM. So this is a mouse now, and the mouse is normal, apparently. It's born normal, and it's still young now, but he shows that he has tissues and DREAM is off. Doesn't work, so it should have better repair. So now he has sent us tissues and asked us to look for mutations. Can we now show that in those dream off mice, the mutation rate is indeed lower than in the normal control mice, dream on? And I just told you, I got this email this morning that our first results, and again, these are sort of, actually, they're not really first results, they're second results. We always do a pilot first and then show it. And we do see indeed a small reduction, which is very clear, in the mutation rate when he in the mouse inhibits dream. Here, there's clear evidence that inhibiting dream is increasing all your repair processes and apparently also reducing, albeit only by a little, mutation frequency. Are we the only ones who found this? No, there is actually a paper, we just found that actually on, well, Bjorn sent it to us, on by Trey Eidecker, also in a mouse model, a different mouse model, and it's on bioRxiv, so it hasn't been published yet, but it's publicly available. And he also finds a very small decline in the mutation rate. So since these are already two independent groups, okay, neither has been peer-reviewed yet, but we find that completely independent of each other in different mouse models. So to me, I'm very excited about it. I don't say it's all true, we still need to confirm, but I'm very excited about the possibilities that we may tinker with dream in the somatic cells, and then maybe not only increase DNA repair, but reduce mutation burden.
Do we know what dream does? And does turning it off have any negative impact?
Wow, that's of course the good question. When Bjorn made his mouse model, he was even expecting that they wouldn't survive. They wouldn't be born alive, but they did, and they seem to be completely normal. Now, why is that? Well, as is usually in this kind of processes, this is very complex. There may be lots of factors that in some way control cell cycling, cell division, and DNA repair. And the fact that what I told you, the effect is really small. So it's like maybe 10%. It's maybe 10% lower, 10, 20% maybe. So we could clearly measure it. It's significant, but it is small. So we don't really know how important that will prove to be for life. In worms, apparently it is, but could it be also in the mouse? Will his mice live longer? You know, this is all very exciting stuff. So you called us in exactly the right moment, because I've never been so excited about this. Because when you asked me this maybe 5 years ago, I would tell you no. I would say if aging is truly caused by somatic mutations, then that's it. We can never fix that. It's not possible. And we will basically always remain stuck at our maximum lifespan. I mean, I don't know, you may be aware of the fact that we published a paper in Nature in 2016, I believe. Yeah, 2016, where we basically argued that human lifespan has a natural limit of 115 years. And we basically derived that from demographic studies on existing data sets. And there was an enormous conversation about that. Because a lot of people were very disappointed. They couldn't believe it because they were so optimistic that we could begin to increase human lifespan, because there's so much talk about neuroscience and everything. And now we sort of threw a bucket of cold water over that by saying, well, forget it, because this is it. I mean, there's absolutely zero evidence that we have improved human maximum lifespan. Yes, we improved mean lifespan. Obviously, we did. People have much more chance now to grow old and stay healthy than in the past. And maybe we can still improve on that, although even I have my doubts about that too. But definitely we cannot break through the limit. Now, okay, to some extent that's also premature, of course, because you never know what will be discovered tomorrow or next year or in 10 years. But for me, it seemed pretty convincing that the only way to tinker with aging itself is by going to the baseline processes, the basic mechanisms. And as you and I discussed now for a long time already, this could be DNA damage and mutations. And if you can slow that down, yeah, then you could probably bring that limit to life up to maybe, I don't know, 130, 150. Where's the limit? We don't know that. that's obviously the well, the pot of gold, not at the end of the rainbow. When we are able to understand what the basic mechanisms of aging are and we can play with it, look, then in a way the sky's the limit, and we are really getting very close to what the old alchemists wanted to accomplish, not to gain immortality. Now, again, I'm not the type to argument. People, most of my colleagues feel that I'm always too negative about aging. That's why they're a little bit worried when I'm going to talk because, well, they think, oh my God, here again, this guy, who's going to say that we cannot live much longer than that, and aging is too complex, too difficult. Yeah, but still, I mean, I would be the first to say, look, let's explore this because maybe we are able to do it. And then there are many other questions that you can ask. Is it desirable to do a thing like that? Ethically, we don't know.
So thinking about these, so there's all these processes, the genes that help with the repair. So I mean, I believe that the PARPs and the Sirtuins are some of these are two genes that help with DNA repair.
Yes.
So just would you think that taking an NAD booster, which PARPs and Sirtuins both need NAD, would that speed up would that increase their activity, and would increasing their activity make any difference to the DNA repair?
Yeah, yeah, no, that's a good question. Now you probably know that NAD boosters they didn't get a good rep recently, but that's almost true for everything. So let's be fair. I mean, anything you come up with, there are always people who criticize it, and although I may have a name to be pessimistic about these things and always negative, I'm actually usually positive. I feel that we need to explore this further. Now, can NAD boosters influence the Sirtuins? Can they boost activity of CRT6, CRT1? Well, I don't know. I honestly don't, and it's not my field. But I do know, and my friend and colleague Vera Gorbinova showed that very clearly, that CRT6 is involved in double-strand break repair. And I mentioned before already how important that is. So it could be that CT6 actually is a master regulator, call it a mini master regulator, a master regulator of only double-strand break repair. Is it? We still don't know. But she found very clearly that long-lived species, they have better double-strand break repair and higher levels of CT6. So here is another correlation. So that tells you that yes, that certainly is an avenue that may be worth following. And sirtuins, known as some sort of longevity genes, could certainly, if we study that the right way in depth, could maybe one mechanism to improve DNA repair and maybe also increase lifespan. Is that likely? I still think not, because there are too many other DNA repair pathways that are not affected by it, and they may then become the limiting factor. I mean, it's always the case. Not you take one possible cause away, but then there's too many others that are still there, and they eventually you'll end up to gain nothing. Now, potentially, that's the danger also with what I just said about DREAM. Theoretically, we could maybe influence all DNA repair processes and slow down all DNA damage and mutations. Now, will that then extend lifespan significantly? Well, it has certainly the potential to do that, but that would imply that there are no other aging processes independent of DNA damage. And is that likely? No. Think about it this way. Suppose now that DNA repair, DNA damage would be the original cause of aging in all species. It would be a universal cause. Then evolution would anticipate on that simply by saying, well, it doesn't talk, of course, but the way I behave now, like I'm evolution, that's how evolution works. Not it selects for things that have effects. It would say, well, if we select for something that let that person live much longer than its mutation rate allows, what's the usefulness? Because the person will be dead. So evolution would never select for something beneficial that would appear later than when after your death. It wouldn't do that. So then, but it was also the other way around. When there are adverse effects, and there would be some gene that is doing bad things late in life or So after you normally would already have died, evolution would do nothing. It would let it sit in. So it means that even if you would neutralize mutations as a cause of aging, there will be a whole bunch of other processes independent of mutations that were not selected against by evolution. They are stuck in your genome. So now you will need to address those processes too. The only hope you can have is that those processes can fairly easily be fixed, while DNA damage cannot. So when we can fix DNA damage, we may go to all the other causes, and they may be relatively simple. We don't know, of course, I'm speculating, but it is a possibility.
Are you aware of any kind of biotech companies looking at mechanisms of improving DNA repair? I mean, so Dream is one, but at the moment you're playing with it genetically, right? So it's not a pharmacological solution.
It actually is a pharmacological solution. You can inhibit dream by small molecule drugs. You can do that by interacting with a certain component of dream that is limiting. And we actually proposed to do research along those lines to begin to use these molecules, these small drugs, to actually inhibit a dream. Yes, you are right. Bjorn did it in the mouse model genetically, but you can do it pharmacologically. But your question was about a company who's doing it. Well, I know only one company that's doing that, and that's also a friend of mine. So I'm not saying that because he's a friend of mine. I'm just this is what I think. He is also a believer, if I can use the word. It's not a nice word actually, but that is sort of, let's say, he's interested in DNA damage as a cause of aging. So he also realizes too that DNA repair must be improved. He has a big company, well, a biotech company, not a big company, of course, but I mean, it covers a lot of ground. Let's say it like that. He his company's name is called Matter, Matter like matter, Matter, Matter Bio, and his name is Christopher Bradley. He's very, very involved in aging research. He's working with us now to do whole genome sequencing for free of our very long-lived humans. Not I say ours, but I mean near Barcelona, my friend and colleague at Einstein, who has collected these centenarians for his whole life, and now we want to do whole genome sequencing, and Chris is going to pay for that. He's doing it for us. But he has also another company. He has three companies basically. One is focused on cancer. The other one is commercializing a new mutation detection method, single molecule assay that we developed. And he licensed the technology from my employer, and he's using that in genetic toxicology testing. But he's also interested in testing it in aging, of course. But the third company is interested because there he's working with George Church and trying to improve DNA repair so that there are less mutations and hopefully therefore less aging. He's not doing that through Dream, not that I'm aware of at least. I don't know the details of this. You need to talk to him to figure it out.
Well, it sounds hopeful anyway.
Yeah, 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 it is.
Um, just a quick thought. So these DNA repair molecules are really important, right? Well, the genes are really important. Are there any SNPs, single nucleotide polymorphisms, associated with them? And would it be wise to get them checked?
Yeah, that's a good point also. Now, people have done that in the past. They try to find SNPs that were in some way predictive for cancer risk, but that was not very successful. And the reason is, again, that DNA repair is very complex. There are so many genes that are important. And SNPs, as you know, are not causal. They are essentially linked, genetically linked to particular genes. And traits, I should say, and it may mean nothing. So that never worked really. Now, what we have done, we have a large NIH-funded grant, multiple investigators, near Barcelona is part of it, and Vera Gorbunova is also part of it, where we try to find rare genetic variants, so not the common variants that people are using that are called SNPs, but the rare variants, in particular genes, that are enriched in the genome of very long-lived humans, like centenarians. So our reasoning was, well, if there is a genetic component, and we know there is a genetic component of becoming so old, then maybe we can see that these rare gene variants can be found in these centenarians, and we can know what they are. And it turns out that not all of them, but many of them are actually in some way involved in DNA repair. And the best known example is a CRT6 variant. We found a genetic variant, again, not a SNP, but it is a real mutation, so it's rare. And it occurs, it's enriched in the centenarian genome, and it actually helps to improve 36. Vera tested that. So we do know, we published that also. So we do know that, yes, at least so some part of the centenarianism, the extreme longevity, yeah, there, you can explain that by rare variants of DNA repair. But and we are now, of course, trying to follow that up and test that in a broader way. But this is really not so simple. Again, one big problem, and we had enormous problems in getting this grant funded because of that, is the numbers. Yes, there are many centenarians now, but not nearly enough. When you talk about GWAS studies and SNPs, people are not talking anymore about thousands of people. They're talking about hundreds of thousands of people. Well, we don't have so many centenarians. So you can see the complexities here. So this is why we moved to rare variants. But even then we got flack for this approach. So we had to come up with lots of other ways how we are controlling for that and so on. But it's an approach. It's something that we definitely do.
Interesting to see whether, yeah, how that will work. Okay, Dr. Vijg, thank you so much. Where can people follow your work?
Oh, well, I have a lab website, but I must confess I pay very little attention to this kind of public relations. I don't do anything like that. I mean, yeah, I have my publications. That's
So,
basically it. I don't like, uh, well, publicity is not my thing. I prefer to do my research with my people, and that's it. And of course, I'm always willing, like this. Now, you asked me for an interview. Of course, I know when we need to do that. We are owe it to the public. They fund our research, so that's why I'm doing it. Obviously.
absolutely understand. Yes, your research is the important thing. So, and thank you very much for sharing your time with me today. It's
Good.
been
Then
very
then I can go out to vote because I need to do that.
okay.
Vote for mayor of New York. Not
Uh,
unimportant, although we
yeah,
know the result already.
not unimportant. Okay.
All
Okay, well,
right.
I will let you go. So
All
thank you
right.
so much. It's been great
Thank
talking to you.
you.
Thank you.
Bye.
Bye.